Welcome to This Explainer. Today, we're unpacking a really urgent and frankly fascinating dilemma that's unfolding right now in the Democratic Republic of the Congo. We're going to look at the hard science, the incredibly intense timeline, and the nearly impossible choices facing health officials on the ground right this minute. Okay, let's dive into it.
Part 1. A rare Ebola outbreak.
291. We are barely 2 weeks into this outbreak, and that's already the number of confirmed cases, along with 43 deaths. The virus is moving incredibly fast, and it's already spreading to neighboring provinces and into Uganda. The clock is absolutely ticking.
Which brings us to a really high-stakes question. I want you to think about this. What do you do when the medicine you have in your arsenal doesn't quite match the disease you're actually fighting? In a literal life-or-death situation, is close enough actually enough?
Part 2. The vaccine dilemma.
To understand why this is such a massive problem, we need to talk about the specific nature of this outbreak. We're dealing with the Bundibugyo species, or BDBV. It's super rare. The surface proteins of Ebola variants differ so much that they are entirely different species, not just slightly different strains. And here's the tricky part. 15 out of the last 16 outbreaks in the DRC were caused by the Zaire species, not Bundibugyo. So naturally, all our best tools are built for Zaire.
Now, an incredible amount of resources are being mobilized. Millions of dollars from the Coalition for Epidemic Preparedness Innovations, or CEPI, are pouring into developing BDBV-specific vaccines. You've got Oxford, IAV, and Moderna all racing to the finish line. Gavi has even set aside $40 million just to guarantee a market for these manufacturers. But there's a tragic catch. These vaccines are still months away from being ready for human testing, and the people in Ituri province, they absolutely do not have months.
Part 3. The mismatched science.
Because waiting is just going to cost more lives. Scientists and health officials are looking at repurposing the tools they already have, sitting in the shed. They're actively debating whether to use Ervbo, which is a licensed, highly effective vaccine by Merck, but designed for Ebola Zaire, or an unlicensed vaccine from IAVI, which was actually designed to protect against yet another species, Ebola Sudan.
And the crucial sticking point here is this 40% figure. The amino acids that make up the surface proteins of these different Ebola species differ by up to 40%. If you think of a vaccine like a key and a virus like a lock, a 40% difference means you have a key that might just barely jiggle in the lock, but there's absolutely no guarantee it's going to turn and actually open it.
However, science sometimes offers us a glimmer of hope. Enter the prime boost strategy. Step 1, you give a priming dose of the Sudan vaccine. Step 2, 14 days later, you follow up with a booster dose of the Zaire vaccine. And surprisingly, in some monkey experiments, this specific combination actually offered full protection from the Bundibugyo disease.
Part 4. The case for caution.
It sounds promising, right? Well, in the medical world, leaping from small animal studies to widespread human deployment during an active crisis is an incredibly risky jump. The World Health Organization brought together their advisory panels, SAGE and TAG, and they are super hesitant. They concluded that there's really only weak evidence supporting this mismatched approach. They explicitly stated it basically neither confirms nor excludes that it'll work. So their strong recommendation is that these vaccines should only be used in strict controlled clinical trials.
And their deepest fear makes a lot of sense. Deploying mismatched vaccines outside of those strict trials could create a totally false sense of security. Imagine telling people they're vaccinated, but then they still get sick and die because the vaccine was a mismatch. You risk completely destroying public confidence in all future Ebola vaccination campaigns. The long-term damage to public health could just be catastrophic.
And even if they do follow the WHO's advice and set up a clinical trial, it's a massive logistical nightmare. As biostatistician Ira Lungany points out, you would need an insanely high number of participants just to detect any subtle protective effect from a mismatched vaccine. In the pure chaos of an active outbreak zone, getting those numbers is incredibly difficult.
Part 5. The case for action.
Now, if you step out of the advisory boardrooms in Geneva and go to the front lines in the DRC, you hear a very different perspective. Local health officials are begging for immediate help. Dr. Steve Ahakamundeke, a virologist at the DRC's National Institute of Biomedical Research, summed up the sheer desperation perfectly. He said, "We have our heads under the water." He is pushing hard to deploy the Ervbo vaccine immediately under compassionate use, right alongside any trials.
And his arguments are incredibly practical. First, familiarity. Between 2018 and 2020, 345,000 locals safely received the Ervbo vaccine. They know it, they trust it. Second, double protection. Even if it completely fails against Bundibugyo, it will at least protect the population against future Zaire outbreaks. And finally, urgency. As researcher Nancy Sullivan points out, using what we have in hand right now is simply our fastest opportunity to save lives today.
You also have organizations like Doctors Without Borders trying to carve out a middle ground here. Their epidemiologists argue we have to immediately test that prime boost strategy we discussed, but specifically on healthcare workers. These frontline workers face massive infection risks, especially when they take off their gear and go home to their communities. Plus, they're way easier to track down for that crucial 14-day booster shot than the general public.
Part 6. A Race Against Time.
So, the scientific community and the DRC Ministry of Public Health are stuck at this deeply painful crossroads. There are two distinct paths. Path 1 is the cautious, evidence-driven route of strict clinical trials. It gets you vital data and protects long-term trust, but it inherently limits immediate access. Path 2 is the desperate humanitarian route of widespread compassionate use. It maximizes immediate protection, but risks a devastating blow to public trust if the mismatch is just too great.
When you zoom out and look at the timeline of this crisis, a really frustrating reality emerges. 16 years ago, early monkey studies were already showing promise for that prime boost strategy. But without funding and attention, that research basically languished. Fast forward to mid-May 2026. The rare outbreak hits. On May 28th, the WHO panels issue their cautious warnings. And now, the DRC ministry has to make the ultimate deployment decision in a matter of days. Decades of neglected research all coming down to a split-second, agonizing choice.
And that leaves us and the health officials in the DRC with an almost impossible dilemma to ponder. I'll leave you with this question to think about. In a deadly, rapidly spreading outbreak, is a mismatched, unproven vaccine actually better than no vaccine at all? Thanks for joining me for this explainer. Keep questioning, keep learning, and I'll catch you next time.